I'd like to thank the organizers for inviting me to talk to you about our work with the gonadotropins. So the gonadotropins, luteinizing hormone and follicle stimulating hormone, play a central role in reproduction, and their connection is between brain activity, which at puberty, the gonadotrope neurons start secreting GnRH in an episodic manner, which causes the gonadotropes to release follicle-stimulating hormone and luteinizing hormone in an appropriate manner that allows ovarian follicles, which have been developing to the secondary stage since early in pregnancy, when the female is still in her mother's uterus. At puberty, these begin to develop antra and approach the point where an LH surge can cause ovulation and transformation of the cyst-like structure into corpus luteum, which is a solid structure. These follicles and corpora lutea secrete steroid hormones, which feed back on both the hypothalamus and the pituitary to regulate the amount and the pattern of gonadotropin release. And it's believed that these steroids affect the glycosylation, although we really don't know how that happens. Glycoproteins are members of the glycoprotein hormone family because they share a common alpha subunit. There's a single alpha subunit gene. And in humans, there are 4 genes, one for FSH, one for LH beta, one for CG beta, and one for TSH. TSH is not a gonadotropin, all of these are. And if you combine FSH beta with alpha, you get FSH. If you combine LH beta with alpha, you get LH. And the heterodimer is necessary for function. These are cysteine knot proteins, which means that 2 disulfide bonds form a ring. A 3rd disulfide bond is formed, passes through the ring, and that throws the subunit into a series of 3 loops. And loops 1 and 3 are on one side of the cysteine knot, and loop 2 is on the other side. The beta subunit has an additional loop that wraps around the alpha subunit loop 2 and is stabilized by a disulfide bond. And by analogy to seatbelts, this is often referred to as the seatbelt loop. FSH and LH are N-glycosylated, so the alpha subunit has carbohydrate at the same 2 asparagine residues. FSH has potentially 2 N-glycans in the beta subunit, and LH has only 1. And while if you look at the proteins, you can't tell the difference between TSH alpha or LH alpha. If you look at the carbohydrates, you can actually distinguish the carbohydrates. So I had a graduate student who used to be able to look at the oligosaccharide pattern from gonadotropins and tell you which hormone, which subunit, and which glycosylation site you were looking at, just by the pattern of oligosaccharides. So we attempted to do some site-specific glycosylation analysis by isolating small glycopeptides and then performing mass spectrometry. Heather Desair's lab at the University of Kansas did the mass spectrometry, but we told her what the glycopeptides were so that she could figure out what once we knew the sequence of the peptides, she could figure out what the glycopeptides were. And there are 4 glycosylation sites in FSH, and the problem is that this method only actually interrogates 2 of the glycosylation sites. Does a horrible job with this one because the glycopeptide doesn't ionize, and not so good with this site. But these oligosaccharides have specific jobs. So asparagine 78 oligosaccharides are associated with folding of the alpha subunit and stabilizing it. Alpha 52 plays a critical role in signal transduction. If the glycan's too big, it may inhibit binding to the receptor. And it may or it may not affect heterodimer stability. The beta subunit glycans appear to play their biggest role in determining circulatory half-life, and that seems to matter because, for example, LH is secreted as pulses, and so it appears in the serum at an hour, 6 minutes later, half of it's gone, and an hour later a new bolus comes, and that's necessary for stimulating the gonads to their maximum amount. You can also have partial glycosylation, and that's hormone specific. So this is an SDS page showing the LH. So the alpha subunits all migrate slow because they have two N-glycans. The beta subunits migrate rapidly because they have a single N-glycan. In FSH, it's different. If you do a Kamasi staining, all you see is one giant smear. But a Western blot allows you to see the reason for that, and that is there are two forms of the beta subunit, a high molecular weight form at about 24 kilodaltons that possesses both N-glycans, and then partially glycosylated forms at 21 and 18 kilodaltons that possess only one of the glycans. And we've been trying to study these oligosaccharides. And so in primates, we see this a lot. In the horse, for example, we see mostly hypoglycosylated. And in the pig and the sheep, we see mostly the fully glycosylated forms. Well, this is an artifact of purification. The partially glycosylated forms are in the LH fraction. In other primates, like Japanese macaque and rhesus monkey, there are at least two major FSH glycoforms. And our studies involve biochemistry and also transgenic mice, and they've taught us that FSH24 has all four N-glycans. So this is the structure of FSH shown as a cartoon, alpha subunit in green, beta subunit in blue. Here's the asparagine 78 carbohydrate, which does not influence function of the hormone. And then these three oligosaccharides, which appear to affect function, are all clustered at the other end of the molecule. So FSH21, which we study a lot, is missing asparagine 24 glycan. FSH18, which when we try to purify FSH21, then the less abundant FSH18 all of a sudden appears, and it's missing the asparagine 7 glycan. What makes these appear to be physiologically interesting is that when we analyze pituitary FSH from women in their cadavers, so these were women who died in their 20s, FSH21 is more abundant than FSH24. In this group, in the perimenopausal period, when infertility is rising, you have a roughly 50-50 mixture of the two glycoforms, and then in postmenopausal women, the FSH24 predominates. And since FSH is isolated from the elderly for the most part, this is the classic picture of FSH, and we didn't notice this high expression of FSH21 until we started interrogating individual pituitary glands. The other reason this may be potentially physiologically important is FSH21 is more active than FSH24. For example, it immediately engages the FSH receptor, whereas FSH24 sort of sits around in solution for a while before beginning to engage the receptor, and then the rate seems to be about the same as for FSH21, but it never binds to as many sites as FSH21 can. If we do classic competition assays, we find that FSH24 is a poor competitor for iodinated FSH, whereas FSH21 can be a lot better, like 10 to 20-fold. And then this is equine FSH, which we sometimes use as a surrogate for FSH21, and it's even more active than FSH21. If we do saturation studies, we find that FSH24 saturates at a lower level than FSH21. So in physiological tissues, the difference is about twofold, and in cells that overexpress the FSH receptor, the difference is about threefold. So this is the FSH receptor, and I have to show you this diagram because there's no crystal structure of the whole receptor. There's a very large extracellular domain that's unique to the glycoprotein hormone receptors, and then there's a classic 7-transmembrane domain that you find in G-protein coupled receptors, and these are G-protein coupled receptors. And in 2005, Fan and Hendrickson reported the structure of the high affinity binding site of the receptor. Sequence had indicated this was probably a leucine-rich repeat protein, and this confirmed that it's a leucine-rich repeat with all the beta strands in the ligand binding site. On the back side, instead of alpha helices, there's some minor beta strands. And so there's no structure of FSH fully glycosylated. To do crystallography, you have to get rid of the carbohydrate. So we went to the Glycam website. that Bob Woods has at the Complex Carbohydrate Research Center, and decorated a molecule of FSH with carbohydrate. And you can see that the protein is interacting with the receptor and the carbohydrates are off to the side. In 2012, Julian Jiang and his colleagues had the structure of the complete extracellular domain, including the so-called hinge region, which was expected to give the rest of this flexibility so that FSH could do strange things like activate neighboring receptors. And instead, it turned out to be a continuation of the leucine-rich repeat, so it's a rigid structure. It does possess this additional loop, which has a highly conserved sulfotyrosine residue that's necessary for activating the receptor. But it's all protein-protein interaction. And sure enough, if you lay that glycosylated FSH model on this extracellular domain, you see again, it's all protein-protein interaction. And if you rotate it so that you can see from the side, here's the receptor, here's FSH, and here's all the carbohydrate on the back side. So the mystery is, if you remove one of the carbohydrates from FSH, the affinity for the receptor increases a little bit. Sometimes it doesn't increase at all, but there's a modest effect. There's a profound effect on the ability of LH and FSH to activate the receptor. So this is LH, stimulating testosterone in Leydig cells, the target cell in the testis. So here's intact FSH, and here's asparagine 56 deglycosylated FSH. Not only does it take 100 times more hormone to stimulate steroidogenesis, you never stimulate the same amount of steroid that you do with the intact hormone. And in fact, you can inhibit, not binding. So this is FSH. Here is the binding to the FSH receptor in the absence of carbohydrate. And here is a hybrid type oligosaccharide isolated from sheep, LH alpha subunit, asparagine 56. And it has no effect on binding. However, it reduces basal steroidogenesis and inhibits steroidogenesis. As you add more hormone, you still never reach this same level of steroidogenesis. These experiments are a little hard to do because here we're adding a boatload of oligosaccharide because of the low affinity when you take the carbohydrate away from the protein. Remember, carbohydrate is delivered by the hormone to the cell by a very high affinity nanomolar Kd protein-protein interaction between FSH and the receptor. In the crystal structure of the complete extracellular domain, a trimer appeared. And Julian Jiang suggested that because the trimer can accommodate in the deglycosylated state 3 FSH ligands, that perhaps carbohydrate was interfering with FSH binding not through interaction to the actual high affinity site, but to the neighboring protein. And so again, if you decorate FSH with carbohydrate, now you can see that the potential for these oligosaccharides to clash with a neighboring extracellular domain actually exists. His attention was drawn to the fact that asparagine 52 glycan, which has long been known to be important for full activation of the receptor, seems to fit in this pocket formed by the 3 receptor extracellular domains, and this presence of this oligosaccharide would prevent other FSH molecules from binding to the 2 other sites. And so when they mutate FSH to remove that oligosaccharide, now all of a sudden the binding goes up 3-fold. So that supported his model. And when he looked at our data, because we're saying, well, you know, with your model, the beta subunit oligosaccharides are still hanging out in space and they're not even clashing with the nearest neighbor receptors. And this is our most active preparation of hypoglycosylated FSH. It's a mixture of FSH 21 and 18. The alpha subunit migrates faster, so he suggested, well, the oligosaccharides, if they're small, you can have multiple ligands fitting in here. And so maybe you're focusing on this, but really it's the size of the glycans here. So it's very difficult to isolate these glycoforms, so the way we do it is we hook 3 superdeck 75 columns together, and then we fractionate and pull out the high molecular weight and the low molecular weight forms to give us FSH24 and then a mixture of FSH 18 and 21. And so we sampled these preparations, these fractions, and asked the question, what's the glycans like? And to do that, we took advantage of the fact that if you use PNGase-F digestion, the beta subunit glycans are not released because of the conformation. The enzyme can't get in there, so when you digest, beta subunit glycans stay on. But asparagine 52 glycans are released. And then we send those oligosaccharides to Oxford, where David Harvey, for the last 15 years, has been helping us by analyzing our oligosaccharides by mass spectrometry. And initially it was MALDI-TOF MS, and then it became nanoelectrospray in negative mode, and now with ion mobility, you can look at, separate out the singly, doubly, and triply charged oligosaccharides. And the problem with this is you get this huge array of data, and how do you make sense of it? So even if you take the approach that the gene array people use, because the charge variants are all over the map in the spectrum, you end up with this horrible mess where you're trying to compare two things. So we stick to comparing the core, the neutral cores. We just sum all the glycans that possess this neutral core and then compare them with one another, and that makes it possible to look at something in a slide that you can understand. Because the sad thing is, we take 10 micrograms of FSH, reduce alkylate, treat with PNGase-F, send off the oligosaccharides to David Harvey, and we get, over the years, we've accumulated 100 different glycan core structures. And if you add sialic acid to this mix, you've got to triple the number of unique glycans that you can have to something like 300. So it's a real mess. So if we just look at the glycan cores, and now we're looking at FSH24, the mixture of the two forms, and then FSH21 and 18, what you see is the most abundant glycans in all of these are actually triantennary glycans. One bisecting is the most abundant, and this is the next most abundant. This is the smallest glycan that's been found in these, and you can see it's absent in the high molecular weight forms, and it's barely present in the low molecular weight forms. This is a pentasaccharide core, and according to Julian, his modeling suggests that a maximum of 4 monosaccharides is all that can be accommodated in that pocket with 3 ligands simultaneously. So even this, which is not very abundant, is too big for his model. When we look at other glycans, what we can see is that when you go to the smaller FSH21, you get more of the biantennary glycans, and only the very largest have a high abundance of tetraantennary glycans at this particular site. So there's a competing model for the gonadotropin receptors. This is the results of super resolution fluorescence microscopy, where they look at the distribution of individual receptors on the surface of cells. And what Kim Jonas and her colleagues have found is that there's an array of monomers, dimers, trimers, et cetera, on up to nonomers. And so here are some of the tetramers, here are the trimers. And in this arrangement, then you could have 2 FSH molecules binding here and here, and they wouldn't really clash with one another. So again, it raises the mystery of what's going on. But we tend to like it because if you do dissociation studies, so you load the receptor with FSH and then you incubate, take away the tracer, the unbound tracer, and just incubate for periods of time, you can go for up to 24 hours and there's no dissociation. If you add 1,000-fold excess cold hormone, now you can drive off some of the hormone, but not all of it. And we're concerned that this resistant hormone was trapped, and so we use low pH to see if we can knock off the hormone, and most of the residual hormone falls off with pH 3 treatment. And of course, these are normalized, so it looks like the binding's about the same. And there's about 50 to 60% of the receptors are probably monomers, and the rest are oligomers. So from Kim's paper, it estimates about 40% oligomerized receptor and 60% monomer. So that sort of reflects the reality of doing these dissociation studies. So perhaps this model is a more accurate one. And we're actually trying to collaborate with Kim to look at the effects of our glycoforms on FSH receptor oligomer number. Hopefully we'll see something by Christmas. So thank you for your attention. I'd like to thank all the people who did the actual work, graduate students like Wendy Walton, Heather Desire, whose lab did the glycopeptide work, David Harvey, who's analyzed oligosaccharides, and my colleagues, Jeff May at WSU, who's been involved in a lot of our program project work, and John Davis and Raj Kumar, the other major investigators. And these are my colleagues, Victor Butnev and Vladimir Butnev, and molecular biologists. And thank the NIH for funding. Thank you for your attention.
Fascinating talk. Really enjoyed it. When you talk about the FSH receptor, which is a GPCR, with aging process, it seems that there is a loss of function. Do you think that loss of function is related to the state of the glycosylation of the receptor?
Receptor glycosylation doesn't seem to affect the function of the receptor. So it's known that receptor occupancy is directly related to hormone activity. And as women age, FSH24, which binds fewer receptors, goes up. FSH21, which binds more, goes down. And so in these older women, you have fewer FSH receptors occupied by the ligand.
And my other question here, have you looked at women that have preeclampsia in terms of this particular study you're doing?
No, we haven't yet.
It would be fascinating to see.
Yeah. We've been trying to switch our assay from pituitaries, which come from cadavers, so they do those women no good at all, to a urinary. And the Western blotting in the urine doesn't work because 300 mils of urine and you have 200 nanograms of FSH, and our Western blots require a microgram.
So it's problematic.
It's very technically problematic.
Yeah, is there any way you can interpret the negative cooperativity a little bit more? In other words, get binding constants, cooperativity parameters, and so on, that might share some light on it?
Well, we're starting to work on that.
Oh, okay. And also, a nasty question. You're sure that the loss of fluorescence means that there's just less fluorophore and not a change in the quantum yield?
Well, this is not my field, but so what Kim does is, the acronym is PALM, so they activate with a laser the fluorophore. So they put an epitope tag on the end of the LH receptor, they take a fluorescent labeled antibody that binds to the epitope, and they activate it with a laser, and then they cause it to fluoresce, and then they bleach it. And they do cycle and cycle after that. And it only does a handful of receptors. So rather than seeing this green glow over the surface of the cell, you get individual positions of the receptors. And then they go through and estimate how many are close enough to perhaps show dimerization. And then with Francesca Finelli, they built the models that I showed the picture of and did docking experiments, and those experiments suggested that you can dock the LH receptor transmembrane domains in a variety of orientations that give you the patterns that you see on the cell. So it's a fairly complicated, sophisticated thing, and involves a lot of modeling, just like we modeled oligosaccharides, and we find they don't interact with the receptor. But they're necessary. We can experimentally remove carbohydrate, and the hormone no longer works. So what's up with that? And that's been a question for 30 years now.
Yeah, my question is on the more basic side. The loss of 21 form is because they are lost, or are they converted to 24?
So when we found the 21 form, we sequenced it, and we found PTH asparagine, which means that no carbohydrate is ever added to the protein. So what we think is happening is there's an inhibition of oligosaccharide transferase. So the precursor is never attached to the FSH beta at all, okay? And we don't know what the gonad is signaling to the gonadotrope to actually inhibit this. We've tried a number of things, but we've never found anything that actually, the first isoforms that were discovered were basically described on the basis of size. So if you overactimize a rhesus monkey, all of a sudden you get all this high molecular weight FSH, as if you're increasing the amount of FSH24. And if you use estrogen replacement therapy, then you get back to a normal small molecular weight pattern of FSH, which suggests that it's estradiol that's inhibiting the activity of oligosaccharide transferase. So we've tried in a variety of systems to mimic this, but none of them have worked. And our latest effort is in transgenic mice, where so they're making FSH, human FSH beta, and we see the three major glycoforms, FSH 24, 21, and 18. But when you give estrogen, you don't see any FSH at all because the levels have reduced to the point where we can't measure them in a Western blot. So either we have to back off on the estrogen treatment, or we have to pool more pituitaries.
I have a quick question. I thought it was interesting that you said that, I guess, the oligosaccharides don't seem to be binding a receptor. The receptor seems to be a leucine-rich repeat unit, right?
Right.
It's interesting because I think there's been work done in this the immune system of jawless fish that these variable lymphocyte repeats are basically leucine-rich repeats that actually act like antibodies, and many of them have been evolved to bind sugars, and like some of them ones that have been isolated actually bind sugars pretty well. I was just wondering if you did any like docking with the structures, the crystal structure of the leucine-rich repeat with small sugars or something like that, because some of these have been shown to bind sugars.
Oh, that'd be an interesting thing to try.
Yeah, it's,
because we've been thinking about, well, are there some accessory receptors that are involved that, you know, the receptor brings the glycoprotein to these accessory receptors that interact with the carbohydrate.
Sure.
But all you have to do to transform a cell into becoming FSH responsive is transform it with the receptor itself. So what are the other proteins that are involved?
Yeah, right.
That'd be a bit of a challenge to figure
Yeah,
out.
I'll show you the papers
Yeah,
later
let's
on.
see that.
Yeah, sure.
Thank you.
Anything else? Okay, I guess we'll move on. Thank you very much again.